Hello, welcome, and thank you for everyone who's joining us. Today we have David Carlson will be talking, who's an assistant professor at Duke University in the Department of Civil and Environmental Engineering and the Department of Biostatistics and Bioinformatics. His research is focused on developing machine learning methods to facilitate data-driven science, primarily using probabilistic and deep learning approaches. He's previously published on a wide range of algorithmic problems, including efficient inference, stochastic optimization, and interpretable models. He's particularly interested in applying these techniques to develop testable hypotheses and design confirmatory experiments, which has resulted in applications in several areas across engineering and health. So with that, I'll let him take it away.
Great. Thank you so much for the introduction. It's a pleasure to be here today. And I should say, just if anybody has any questions during this, I have the chat open on my other screen. So please just feel free to type in a message to the chat or just unmute and ask a question. Happy to take questions or comments whenever. So I really have fun with this first slide. And my goal of this talk is to make you understand why this picture that I have here that's clearly AI generated is actually very relevant to the problem at hand. So I wanted to start out by just talking about a little bit about what is a brain state. So a lot of people haven't heard of a brain state before. So in this talk, I'm going to be talking primarily about animal models and mostly mice. And so a mouse has a brain, just like a human. And what happens in this brain is that there's a lot of things going on. So there's a lot of processes that are happening. And so when we talk about brain state, it's really a very broad kind of description of what the general activity of the brain is. And in particular, what we are so very interested in is how does this link to broad patterns of behavior? So not necessarily so much about what are you going to do immediately, but more what's your general mood? What's your general, something like an emotional like state? So we might say that there's a broad pattern of activity that's happening in your brain right now. And it might say that you're in a happy mood. You might be a social mouse. You might want to be nice to people and talk to them. Alternatively, it could say that you're in a pretty angry mood and that you're actually going to be a very aggressive. And this might be expressed inappropriately, right? This may be pathological aggression. And so a lot of what we want to be able to do, and a lot of what I'm going to be able to talk about today, is this idea that we want to be able to come up with ways that we can actually take brain data, actually measure it in real time, and if it looks like we're going to inappropriately express aggression, we want to be able to actually intervene. So we actually want to be able to say, this is your current brain activity. Now we want to be able to intervene in real time and actually say, let's stop the inappropriate aggression and restore a more normal state, or perhaps a more friendly, sociable state. So this probably sounds a little bit wild, and there's really this part of this where I'll give myself a thumbs up, great, thanks Zoom. I'm really a machine learning researcher, and so this seems like a little bit of a strange thing for me to be doing. And so there's this really big question that I want to get through here, which is that how does machine learning learn brain states and design interventions? So if we're talking about something like a brain state where I'm telling you that we want to be able to measure the broad patterns of what behavior is going to be in the future, and I'm telling you that we want to be able to actually intervene, how can we actually do this with machine learning? How can we actually solve these types of problems or approach these types of problems with modern AI ML techniques? And so my research group, we use a lot of machine learning to learn brain network models of emotions and behaviors. And one of the things that's going to be really critical here is that we need to be able to explain how we can make our predictions. So if we say that we're going to have a sociable mouse in the future or an angry mouse in the future, we need to be able to explain what's happening in the brain in order to actually make that prediction. And we need to be able to explain it not just to me, but also set it in rules that can be explained to neuroscientists. And by using this fact that we can use explanations and we can have explainable AI, the idea is that we want to be able to generate testable hypotheses. So let's say I find a network related to aggression. Then I actually want to be able to design an intervention and actually change behavior by implementing this experiment. And so this is this idea that I like to talk about a lot, which is that we want to use explainable AI to find testable hypotheses. And from that, we actually want to design confirmatory experiments or interventions. We actually want to develop hypotheses, and actually go through the experimental design to test them. So pretty much everything I'm talking about today is run through the Collective for Psychiatric Neuroengineering. The Collective for Psychiatric Neuroengineering is co-led by myself and Professor Kafui Daraza. Kafui Daraza is a psychiatrist and neurobiologist and biomedical engineer, and he's doing a lot of the animal work here and a lot of the implants, whereas I'm doing all the computational modeling. So this is a very big collaborative effort where we're combining a broad range of expertise in a very interdisciplinary project. And so kind of just to reiterate this approach that we're going to take today is we're going to have some sort of implant, which you can see here. This is an implant directly into a mouse brain. And what we're going to be able to do is we want to take AI and machine learning techniques, and I'll get into what these AI and machine learning techniques actually are, and we want to use this to summarize brain activity. And in particular, we want to find a brain network. And we are going to be finding something called an electrical functional connectome, which is finding patterns of electrical activity in the brain. This looks pretty complicated. I'm going to break down exactly what this is in just a few slides, but for now, you just need to know we're using machine learning and AI to find some sort of network that is related to brain activity or behaviors of interest. And from that, we actually want to say, well, if this is the brain network and how strongly we express this brain network is related to behavior, that actually means that we should be able to design or choose an intervention that manipulates this network and actually changes behavior. And we want to do this in this kind of closed loop here, right? So this is something that we're going to use AI and machine learning to design experiments and actually run them to actually test our hypotheses and directly manipulate the brain activity. This is an idea we've been working on for quite a while now. So the references I'm giving here are actually from many years ago. One of them is in biological psychiatry, and the other is in cell, where we are using these kind of and piloting these types of ideas. So for those of you that aren't familiar, the data we're looking at here is something called a local field potential. So when we implant electrodes into the brain, which is shown on the top right here, we're able to capture electrical signals. And in particular, we're going to implant into many different brain regions here. So what we're showing here is that we can collect a lot of different brain regions, and you get a lot of these kind of waveforms and these squiggles that happen through here. And these are going to capture brain waves. So if you were to take some of this signal, you can break it down into your canonical brain wave bands. So for example, you could break this down into the delta band, you can break this down into the theta band and the beta band and the gamma. And the idea is that these patterns and how strongly we're expressing these frequencies are going to be one of the things that's very important to try to relate to behavior. The other thing that's going to be very important is that we want to understand how the brain is communicating. So what we want to be able to do is we want to be able to look at relationships between brain regions. So one of the things that's going to happen is if two brain regions are communicating, we expect them to be synchronized in some way. They don't have to be exactly lined up like we're showing here, but we will expect that there to be coherence. So if you have coherence and that there's a statistical relationship between two brain regions, we assume they're communicating in some way. Whether that's directly or indirectly is a little bit hard to capture, but we know that there's communication happening. And one of the things that we actually want to figure out is also not just is there communication between these brain regions, but can we tell which way is a directional flow of information? So in the example I'm showing you here, we are looking at two brain regions. We're looking at the hippocampus, which I have up here. And in the bottom, we have the prefrontal cortex. And what you can see is that the peaks of these signals are kind of matching up, but it looks like the hippocampus happens first, and then the prefrontal cortex kind of follows the same signal. If you were to see something like this in a particular frequency band, this would be a sign that the hippocampus is sending information to the prefrontal cortex. And this is defined by something called Granger causality. So it's this weak form of causality, and it's not really causality, it's more just a correlation that looks and suggestive of a potential causal relationships. But one of the things that we can say here is that it looks, when you get data like this, that the hippocampus is sending information to the prefrontal cortex. And vice versa, it might happen the other way. So we might be able to look at this frequency band, and we might be able to go from our hippocampus here, And we might be able to say, well, it looks like the hippocampus is lagging the prefrontal cortex. So we might say that if we were to see a scenario like this, it looks like the prefrontal cortex is sending information to the hippocampus. Now, obviously, we're not going through and looking at this visually for all of these different frequency bands we might look at. We are doing this computationally, so we can look at all the different frequency bands and all of the different pairs of brain regions at once. But one of the things we wanted to be able to do was to say, well, can we actually find, if we have all these different brain regions and we have all these pairwise relationships and we have all these frequency bands, can we actually find some way of describing the important part of the behavior? And so one of the things that we came up with was this idea that we can describe neural activity and we can find the part of neural activity related to something that we care about, and we can describe it as an electrical connectome or an electome network. So I want to spend a minute here explaining what this is. So what we're doing here is we are actually finding patterns of behavior that are related to something that we want to study. So in particular, here's a brain network that I can tell you if it's increased expression, that it's analogous to stress. So this is an animal model, so it's hard to tell whether this is actually stress or not, but we can say that if we put an animal into what would be a stressful situation, that this is a network that is increased. And the way to read this is that we are showing here in this Circos plot over here, we're showing the different brain regions that we have measurements from. So for example, we have the ventral tegmental area and the basolateral amygdala. And what we want to be able to do is we want to be able to say, well, what are the patterns of importance for trying to study this behavior? So if you have kind of these marks on the outer circle, this is saying that there is increased power when you're experiencing this behavioral condition in the frequency band here. So this is 10 hertz, 20 hertz, 30 hertz, 40 hertz, 50 hertz. So this is saying at 5 hertz or thereabouts, you're experiencing increased power. And we can also look at the connection. So if you see a connection between two different brain regions, this is a sign that these two brain regions are increasing the amount of communication they're doing when you're in this scenario. So one of the things that we want to be able to look at is just to say, can you find the patterns that are actually related to this? And so if you're experiencing stress and you're a mouse, this is something that goes up, where you can say that you have this broad increase of power and coherence at 5 hertz. And then what's interesting is that if we look at this 15 hertz, there's an increase just in coherence. So one of the things that happens is we don't really increase power at 15 hertz, but we're increasing coherence at 15 hertz or thereabouts. And I should say the color here doesn't mean anything. The color is just helping us identify different frequency bands so that we can look at them easier. And one of the other things that we can do is we can look and break this down by which regions are leading and which regions are lagging. So we can actually go through and just say, well, which regions seem to be sending signals out to help drive this network? And so we can say that, well, it looks like if we look at our two different frequency bands, that the infralimbic cortex, the nucleus accumbens, and the prelimbic cortex are all kind of sending signals out to help drive this network, and they're doing it in different frequency bands. So this is the basic idea. We want to be able to find this brain network that is related to the things that we care about. Now, there's this really big challenge, which is that the brain doesn't do one thing. The brain does many things, right? I'm currently talking. So what happens right now is I'm in a reasonable mood. I'm fairly alert. I'm talking. I'm thinking about the next slide that's going on. I'm trying to watch all these things. The brain is doing many, many different things at once. And also you can experience many kind of different general feelings at once. And so kind of the challenge is that we might have many different electome networks or many different brain networks to express a lot of different things that might happen. And it's really hard to get and probe exactly what is happening in these animals because they can't self-report, and even self-reports have some challenges anyway. But we want to be able to look at these different brain networks that are happening in all these different cases. And in fact, if we're trying to find something related to, say, stress or social ability, we need to be able to account for the fact that you're feeling many other things at the same time. And these other things are variable and fluid through time. So the idea that we came up with is that we want to have some sort of superposition model. So we might want to be able to model brain activity, which is just to say that we want to take all of our brain activity here. And what we're going to do is we're going to say that we have many different brain networks. So I'm only showing 3 brain networks here. In practice, we tend to have many, many more brain networks whenever we're actually fitting these models. But it's this idea that we have some sort of brain network, and we have another one, and this one is a third one. And the idea is that what's going to happen is we're going to model the brain activity at the current point in time is this first brain network, and it gets to have time varying expression. So we're saying here, XW is X window W, and we're saying it's made up of our first brain network times SW1. This is just saying that there's some sort of network expression that varies as a function of window and as a just the brain network you're looking at. So we're going to have some sort of different score or different expression for our first brain network, our second brain network, our third brain network, and this will vary through time. So for example, this is our network here that was related to stress. We might say that if in a stressful situation, we would expect SW3 to go up, and if we leave a stressful situation, we expect it to go down. And so we're going to try to model these things as a function of windows, where we're actually going to say, how is this actually relating and modeling these things over time? So for anyone that has kind of a background in computational models or statistical models, this is really done through a factor analysis approach. So just to kind of briefly explain what's going on mathematically, what we're going to have here is we're going to say that we are going to have a window, and our brain activity is described here. And we're going to say that we have some sort of brain networks here. So we're going to say k equals 1 to k, where k is the number of brain networks we have. We have our network expression, which is just how strongly that brain network is expressed. And then we have some sort of vector that's going to represent this network. And this is a challenge if we're going to fit it, but this is a thing that's been done for a very, very long time. The most famous factor analysis model is really probabilistic PCA, which the only difference between PCA and probabilistic PCA is that they put this error term on there. Otherwise, probabilistic PCA and PCA are exactly the same thing. But it's this idea that we're going to be able to do this dimensionality reduction. We have a big complex signal, and we're going to map it to a bunch of latent activities or network expressions. So we need to modify this just a little bit for our case, right? Because we need to be able to learn networks. Now, there's a couple tricky things here. One is we're making an assumption that we need to learn non-negative decomposition, or in other words, we're going to assume that a network can only positively express. And so kind of the consequence of this is that saying rather than have one network where you might be happy or sad, depending on if it's positively or negatively expressed, this means that you have to have a network for happiness and a network for sadness. And so that's kind of the assumption that we're building in there. But it also needs to be able to capture these spectral properties and these kind of directional or communication properties that we said that we care about when we're trying to find these electrical functional connectomes. And so we created an approach where we can do this, and it looks very, very similar to a factor analysis approach. Technically, this is a functional factor analysis approach because what we need to be able to do is map from the spectral properties that we care about into kind of the observed space. There's a lot of complicated math that happens in here. The concept itself is rather straightforward, but I'm not going to go into the math. I have the representations down there, the references, I should say. So there's this kind of statement I will make, which if I needed 2 NeurIPS papers to explain it, it probably is pretty complicated. And it's not too bad in terms of concept, but actually to implement this and get it to work took a fair amount of effort. And so there was a study that we initially used this on that I'm not going to talk about too much because this was several years ago now, which is that we were actually able to, the first time, actually use this in an animal model of depression. And we were able to show that we were actually able to find a biomarker of susceptibility, meaning that we were actually able to find a pattern and a brain network that actually signified whether you were actually susceptible to depression or not, which is actually rather exciting because it means that we were able to potentially predict who can get depression, but also try to come up with ways of preventing it. And this was in Cell many years ago, and there's a lot of information there. that I'm not going to go into because I want to talk about some of the things we've done more recently, which is that we wanted to try to extend this to try to look at social behavior and aggression. So I'm going to start out by looking at social behavior. And it's this idea that we wanted to be able to take this exact same approach that we did, and it's very successful for looking at depression vulnerability, and actually really helpful in trying to find biomarkers that were actionable and we could actually work on for depression. And we wanted to say, well, can we do the same thing and try to use it to explain social behavior? Because social behavior is heavily disrupted in a lot of neuropsychiatric disorders, along with a number of developmental conditions. Right. So there's a lot of different disorders. So, for example, social activity gets messed up in depression. And it's also different in children, for example, that are identified with autism. So it's implicated across a large number of conditions. And we wanted to be able to say, can we actually take that same approach and could we actually use it to try to find the social network, a network that could tell us whether we were looking at a social action or just some sort of object exploration. And the reason we're pairing it here with object exploration is the fact that we wanted to be able to kind of disambiguate the fact that you are both exploring and interacting when you're having a social engagement. So it's trying to pair a more similar condition where you're exploring in both cases. And one of the things that was rather unfortunate and actually rather surprising to me is that we took this unsupervised approach that was actually successful, not just trying to find a depression biomarker, but actually in a number of cases, and it actually failed. We went and we tried to do this, and it actually did not work at all. It basically was meaningless. And so we had to think very hard about this because one of the things that we really wanted to be able to do was to be able to find a brain network so that we could actually manipulate the brain network and use it to design interventions, which I'll get to in a second. And so one of the things that was key here is that these type of unsupervised learning approaches, where we're just trying to model like the covariates, it works really well when the data variance is driven by a behavior of interest. So it turns out things like stress and depression are very strong signals that you can isolate pretty well. And in comparison, the social brain signature actually explains a small amount of variance. So if you look at the total amount of information explained about what's going on while you're engaging in social activity, it's actually pretty small. So it's a fairly low signal to noise ratio. So because of this, we really needed to modify our models to include supervision on the model. And what I mean by there is that we needed to guide the representation towards the needed outcome. If we needed it to represent the social behavior, we needed to explicitly require that it encoded social behavior, not just trying to find networks in an unsupervised fashion. And the other thing we needed to do, especially as we are trying to map this in a real time to try to actually look at, say, can we make interventions, is to efficiently approximate the solution. So we need to do this computationally efficiently. So what I'm showing you here is basically the concept that we came up with, which for anyone that has a background in statistics, this is really a supervised factor analysis, where it's the idea is that if we look at this left-hand part over here, this is our unsupervised part. So it's still the exact same thing I showed you before, which is that saying that we have these networks, they get multiplied by some sort of time-varying network expression. And we're going to use all of our brain networks to explain our current electrical activity in the brain. However, we want to actually also require that our network expression is going to be predictive of behavior. So this is an interesting thing to require. And also you may notice that if we look at this left-hand side, we used all of our brain networks to explain what was going on in the brain. However, when we are trying to do the supervised part, we are only going to use one of our brain networks in order to try to explain whether we're looking at a social or a non-social behavior. And the reason for this is that we actually think that this is going to really help us when we're trying to design interventions. Because what this is going to say is that we're going to find one social network. And so that's going to allow us to find a single network that we can use to act on and actually try to design this. So this is an atypical structure where we're going to use all the information to explain our observed local field potentials, our brain activity, but only a single brain network is going to be used to describe the behavior of interest. This can, of course, be generalized. You can use 2, 3 brain networks, but you really want to reduce this because it's going to help us with our ability to explain the key results of the model, but also trying to use this to explain what's going on and design interventions. So the other thing that we're going to do here is that before we were using a statistical technique, so we were really using essentially an expectation maximization to try to infer our latent expression. And now we are going to use a neural network. So this is really a supervised autoencoder or a supervised variational autoencoder in order to try to do this. And the reason we're using a neural network was originally because we thought that we could do a really fast approximation of a neural network. People complain about neural networks being slow, but they're usually slow to train and then very fast once they're trained. And what we really need is that when we're deploying this can be run very quickly. But one of the things that we found is that actually a supervised autoencoder worked much better than a supervised factor model. And it turns out that this is actually a rather surprising result. And because I need to get into all of our biological explanation, I'll give you the one-sentence description, which is that it turns out that this works a lot better than a supervised factor analysis model that is when you have small variance, right? So if you're going to predict the outcomes with a small variance term, like social behavior was, it turns out to be a much more robust and much better system than actually the traditional statistical estimation technique. And I'm not going to go into it more than that. For anyone that's interested, this is Talbot et al. This is actually at the Journal of Royal Statistical Society, and it's about a 40-page paper to explain what I'm saying there. So it turns out that the supervised autoencoder actually has some really interesting statistical properties because we're only going to use XW, which is our LFPs, to explain our latent state. Anyway, so after making these changes, we were able to take this exact same approach, and we were actually able to use it to find a brain network, the social brain network, that could explain whether you were looking at an object or a social interaction. And so this is the brain network here. So I'm going to show you this just very briefly, where I'm showing you on the left, these are our power patterns across the brain regions we measured. This is the directionality that's shown here in more detailed form and then in a more simplified form on the right. And one of the things that we wanted to be able to do is just confirm that if we look at the network expression here, so how strongly this network is actually expressed, do we actually capture behavior? So what I am showing you here is that I am showing you EN social activity. So this is that network expression. So if it was highly expressed versus lowly expressed. And these are all on holdout animals. And what we're doing is we're saying here is we start an interaction and we look at the social engagement, which is shown in green. What's going to happen is that as you start the social engagement, you're on average going up and you're having a much stronger expression of this brain network. And as the interaction ends, it's going to go down. And in fact, it actually goes down in primes before you actually do the interaction, which is kind of interesting as well. And on the bottom, we have some sort of object engagement. So we can find a brain network that tells us whether we are looking at social engagement or an object engagement. And so I can make a claim that this is a social network and that this is controlling social behavior. And if you go and you tell a neuroscientist, hey, I found the social network because it can predict whether I'm looking at social activity or not, they are going to come back to you and basically say, well, you didn't prove that it actually could control social behavior. All you did is show that this is a biomarker. And then you showed that you can predict this. It's not the same thing as saying that we found a social network that is controlling actually what happens in social activity. So there's this really big question we have, which is that we devised a very complicated statistical technique to find brain networks. And we have a lot of experimental data in order to try to find the social brain network. And we need to actually now try to figure out, does this actually control? social behavior or not. So one of the things that we like to look at is this kind of scales of generalizability. So if we think that something is a fundamental brain network, so it's some sort of brain network that controls social behavior, we want to start looking at it across scales of generalizability. So one of the first things you want to do is, if you think this is a brain network related to social ability, you want to show that this holds up in new animals, so new subjects, right? You can also say that if we have this, we should actually have this generalized to new tasks. So we have our original task. If we try a new social task, it should also work. But there's also this thing of, there are a number of drugs and other sorts of manipulations that are known to change behavior. And if we change behavior, does this also change brain expression in the ways that we forecast? We think that it should hold across stress, across sex, across strain. And then as we're getting very ambitious, we are thinking that we should have this hold up across species, which is going from mice all the way up to humans. And I should say, we actually are starting a pilot project where we are starting to look at depression in dogs. So this is something that we are working towards, but I'm not going to talk about that today. And so one of the things that we wanted to do is to say, well, if we think we found a social brain network, can we manipulate neural activity in the ways that it says, and actually use this and then evaluate if we forecast a brain intervention and we say it controls social behavior, can we actually run this brain intervention and then actually measure, do we actually change social behavior or not? So there's a very popular technique, which is called optogenetics. You can use a lot of things. Basically, the long story short is you can actually change the neurons such that they are susceptible to light, and then you can trigger stimulation in the brain that will make neurons fire. Now, you might have a question here of, I've been talking about local field potentials, and I just said that there's some sort of ways we can make neurons fire. So it's really important to understand the context here, which is that this neuronal activity is really averaged over a lot of different neurons in order to get up to that population level activity, which is that local field potential that we're talking about. So when we come up with our lectome model, it's really capturing this population level activity, which is really made up of a lot of different neuronal activity. So the idea here is that even if we're designing the therapeutic based off of what we get in our lectome model or LFPs, if we're applying it to neurons, we still think that it should loop back and change our lectome expression. So we have this hypothesis, which is that our EN social, which is our social lectome, we have a hypothesis that if we were to increase it, we would increase social preference. So this comes up with this idea of, if we have this social network, can we upregulate this network? So we have this network that we found, we've described this, and then it turns out that we can actually choose particular brain regions and connections between brain regions in order to manipulate and try to change what's going on. And so what we are going to do here is, the arrow got slightly off when I copied it. It should go from the prelimbic to the nucleus accumbens. But basically what we said is that if we actually find this, we can look at this connection here, and we can actually say, well, what seems to lead this brain network the most is the prelimbic cortex. So we're going to stimulate brain neurons that are coming from the prelimbic cortex out to the nucleus accumbens. And we're going to stimulate at 10 hertz because that's what we found was important in our model. We wanted to be able to actually do this and stimulate by what we found. And so there's this idea that if we stimulate an important terminal and where we actually start sending signals from this brain network, that we should increase the expression of this brain network. So what we do when we do optogenetics is that we try two different things. So just by simply the fact that you're putting a laser into the brain, it is going to change heat and going to change other things. So you always pair it. So what we have here is we're going to use either a blue laser or a yellow laser, and this is showing the way we randomize the order of the experiments. But the blue laser is going to cause the neurons to react, and the yellow laser is a control. So what we're showing here is that if we look under this yellow laser, which is again our control condition, during an object interaction, versus when they're in social engagement, basically it's just a random change. So if we look at how much stronger this changes things under this yellow laser, it doesn't do anything. Whereas if we actually were to use our blue laser and actually stimulate, we get a dramatic increase in our electron network activity. Now there's this question of, okay, great, so we can increase the activity expressed in this electron network, but we're basically targeting a key terminal and upregulating it. So there's this question of, does this actually link to changes in behavior? And so the answer is yes. If we were to actually go through and do this stimulation here, it actually will enhance social behavior. So one of the things that we want to say is that if we think that we are actually going to look and say increase social behavior, we want to make sure that we're not just increasing exploration. And so we check whether increased social behavior by the amount of times an animal spends socializing. And the answer is very strongly yes. And then we also check, does it change the amount of time that an animal spends looking at a box? So these are all randomized, and the yellow is the control condition, and the blue is the experimental condition. So what we're finding here is that actually by doing this complicated machine learning approach and designing our stimulation to follow what we discovered from this ML approach, we were actually able to manipulate and change the brain expression, but actually control behavior. We're able to increase social activity without increasing exploration in general, which is pretty cool. So one of the things that also is pretty important here is that stimulation frequency matters to outcome. So there was a paper that actually stimulated at 20 hertz, and they actually found a negative change that actually decreased social behavior. So this was actually a pretty surprising thing, because we stimulated at 10 hertz and another group stimulated at 20 hertz, and they didn't find the same changes we did. And so we actually reran this experiment where we stimulated at 20 hertz, which was not in our electron network. And we found that actually stimulating along not just the brain regions that we found were important, but also the frequencies we found were important. Turns out to be very important for changing behavior. I'm not going to go into this too much. I should say there was also a great paper out of Stacy Bilbo's lab where they actually show that this network expression is also disrupted in an air pollution plus maternal stress model of autism, which is pretty interesting as well, and also relating it to microglia. So where I started this talk and where I kind of opened was more on this idea of, can we infer and change brain state as necessary in real time? So not just can we actually say, change whether an animal is being social or not, can we actually infer and change brain state as necessary in real time? Okay, so this is the idea. We want to be able to tell, when we look at an animal here, if it looks like it's going to experience inappropriate aggression, can we actually stop, make an intervention only as necessary, and change its brain state so that it's, again, a more sociable, happy mouse? And so this is what we're going to do next. And I have about 10 minutes left, so I'll be able to explain some of this. And so we have this assay here. And so what we're going to do is we're going to look at three conditions where we're looking at interacting with a male mouse, a castrated mouse, and a female mouse, where you would expect really aggression in a typical case, only in the male-male case, because they don't want to be aggressive towards females in general. And so we're going to actually look at attack behavior. So we're going to define attack behavior here as that biting, boxing, tail rattling, tussling. So kind of actually attacking, a non-attack behavior as sniffing, grooming, or resting. So there's a whole lot of context I'm not going to explain here, but I wanted to say we actually found an electron network that was very good at predicting aggression in general, but actually it was flipped. So it was actually aggression and inhibition. So actually this was an electron network where when it's high, you weren't aggressive, and when it was near zero, you weren't inhibited to aggress. But this was this brain network that we found by training on the same assay that we showed before. Now, this is the big question here, which is, if we have this, and we have this brain network, and we can map to it in real time, can we actually use this in the same way we did before? Well, the first thing we wanted to check was before we start looking at interventions, we wanted to ask, does this generalize? So the first thing we did is we wanted to check to make sure this relates to neural spiking. This is a big thing in the field because neurons are considered the fundamental units in the brain. So if you're manipulating fields, you want to actually show that they fundamentally relate to neurons. They do statistically relate to neurons at a pretty high rate. But we also wanted to look at, say, if we actually change the paradigm we're looking at, rather than looking at actually being attacking, can we actually use other assays and actually get that this brain network and this activity that we found, can we actually get that this holds up in different contexts? So we actually did a urine assay. So basically it's urine from a competitor. And if you put them in an environment with a urine, it's known to increase expression because they're having to smell one of their competitors. And we actually show that this changes this EM inhibition activity as well. And we also want to do this we can actually use designer drugs that were known to increase aggression. And we can actually say that if we use these designer drugs, they also, again, manipulate our brain network activity. But where I want to spend a lot more time is on this concept of controlling aggression with optogenetic stimulation. So one of the things that we came into this knowing is that there was prior work that direct stimulation of the infralimbic cortex is known to reduce aggressive behavior. And this is done at 20 hertz. So if you stimulate in the infralimbic cortex at 20 hertz, you're actually able to decrease aggressive behavior. So this was already known. And so we went through and we actually checked this, and this actually holds up. So one of the things that happens here is that if you were to do this sort of stimulation and you were to use this in aggression, you have that when you actually look at the yellow, which is the control condition, versus the blue, if you look at the amount of time a mouse spends aggressing, doing this neurostimulation dramatically reduces how aggressive a mouse is. And that's great. That's what we want. But there's a big problem that you have a lot of off-target effects. So it also greatly decreases how much time you spend socializing, which you can tell by looking at how much time it spends interacting with the female mice and also a castrated male mouse. So really kind of the message here is that this neurostimulation, this optogenetic stimulation that's done here is not just relevant to aggression, but it's actually relevant to all social behavior. And for anybody that doesn't know, aggression is still considered a social behavior. It's really antisocial, but it's in the family of social behavior. So what actually happens here is if you do this sort of stimulation, it just decreases all social behavior. So we wanted to ask a question, which is, well, if we find our general network expression, can we stimulate only when necessary? So we have a machine learning model. It's running in the background. We can run it in real time by doing this fast mapping through this neural network. We have about 200 milliseconds of lag between pulling out data and estimating our brain state. So it's really kind of minimal lag. It's pretty fast. So it's this question of, well, if we're going to be able to do this in real time, we want to be able to have a mouse. We want to be able to use this network encoder, and anytime it looks like this mouse is going to be aggressive, we then want to stimulate. So we want to be able to take this and actually use this information and then do stimulation only when necessary. And only when it looks like it's going to engage in aggressive behavior, not any social behavior. And so what we wanted to first check is that, well, if we actually do this closed loop stimulation, does it actually change our network expression? And the answer is yes, dramatically. So our network expression is changed tremendously by doing this stimulation. And because we're only stimulating sometimes, we can just check how long after stimulation it takes to change this. And basically, a second after stimulation, you really ramp up and you really get pretty high levels of this aggression inhibition network. And the thing is that's great here is actually we get a very great reduction in how much aggressive behavior you have. So across the board, every single mouse we tested here decreased the amount of time it spent aggressing. But one of the things that's really nice here is that because we're only stimulating when necessary, we don't stimulate as much. We do stimulate some, but we don't stimulate as much when they're engaging in pro-social behavior, so engaging with other mice that are friendly. So if we look at the impact on how much time they spend engaging with a castrated mouse or a female mouse, we see no significant changes. So the big idea here is that if we can stimulate and use these machine learning methods in real time to stimulate only when we need it, we're actually able to change just the aggressive behavior, which is pretty cool. Now, one of the questions that comes up a lot is that, say, is there just something special about the temporal pattern? So rather than stimulating a fixed pattern, we are now stimulating in kind of a more kind of specified pattern. Is there just something special about this pattern? So the way we check this, and this is a standard thing to check in a number of studies in a related area, is you want to do sham neurofeedback in some sense, where basically what we do is we take mouse one, we take its recording and it's time locked, and then we stimulate mouse two based on the stimulation pattern from mouse one. And there are real reasons for including this. In fact, in humans, in neurofeedback studies, they actually find that when you do this, you get a huge placebo effect. So this is an important thing to check. And what happens here is that when we do this non-synchronous stimulation, so we keep the temporal pattern, but it's no longer linked to the mouse's brain activity, we don't see any change in aggression. So it turns out that actually we do really need it locked to an individual's brain activity. Okay. And so I'm going to wrap up here in a minute or two. So I want to get through one more thing, which is just, is this network or this internal state causal? So what we wanted to do is we actually wanted to say, well, we stimulate, we showed that when the stimulation happens that we change our network expression. And then we also show that this changes behavior. But there is this question of, well, does that actually mean that it's causing it? So, or is it just the fact that network expression is somewhat reflective of what's going on in behavior? So the way we tried to go about doing this is by using mediation analysis. So what we're going to do is we're going to run a mediation analysis. This is a causal inference technique where it's the idea that you get some sort of independent variable. Here, this is whether we're stimulating or not. And in particular, this is our blue laser versus our yellow laser, I should say. And we're going to look at our mediator variable, which is our network activity. Let me actually put it here, which is our network activity, and then we have our outcome behavior. So it's really this idea is that, well, can we actually use the statistical approach to actually say, do we have to change network activity to change the outcome behavior? And the answer is yes. So we can actually go through and say that we seem to only get good changes in the outcome behavior when we actually have good changes in the network activity. So we actually need the network activity change to control the outcome. So actually this is some evidence that yes, this brain network is in fact causal and controlling the aggressive behaviors. And if we look at the direct effect, which is basically how well you can explain the outcome, the direct effect isn't, its P is greater than 0.05. Now I will give a caveat here. We're only so well powered here, so there's probably some direct effect that we're not capturing in our brain activity, but there is pretty strong evidence that this brain activity is at least partially controlling the aggressive behavior that's actually happening. So just to conclude, one of the things that's really cool here is that, or at least to me, I'm probably biased, is that we can identify a brain state and actively choose a manipulation in real time. And by actually finding this, by finding an aggressive brain state, it opens up a lot of avenues to actually try to aggress and suppress aggressive behavior, but not impact pro-social behavior. And so right now we're actually working to extend this to other conditions. And we are working a lot on anxiety. We're hoping to have a preprint out on anxiety in the near future. And we're also working on reward progress or goal progress, which is related to how the brain processes reward and also addiction as well. And so I should say also, I am one of the leads on the Autism Center of Excellence. And through the Autism Center of Excellence, we are trying to use similar mathematical techniques to help understand how autism progresses in children diagnosed with autism. And there, we're definitely not making any interventions. There's just a statistical analysis of trying to understand brain development, but we're using a lot of the same mathematical techniques. Because this is an internal talk, or mostly internal, what I did want to say that there's a couple of methodological thrusts that we're working a lot on and continuing to work on that are of a lot of interest to me. So if people are interested in these, I'm happy to talk about that. One of them is domain adaptation and generalization, which is just this idea that if we want to adapt to new scenarios or new subjects, we can actually use a lot of these techniques that have been developed in machine learning, including by our own research group. I want to get better features of directionality, and this is actually a very complicated thing to do because it's a lot of complicated signal processing, and we've been working a lot on that. And we want to be working a lot more on causal models, and we've been working a lot in causality recently, not so much on trying to improve fundamental causal theory, but actually in the sense of how do we extract out brain networks that are actually causal? How do we incorporate causal inference while we're doing this? With that, I should say this was joint work for the Collective for Psychiatric Neuroengineering, co-led by Koff over there. There's a number of people that have worked on this. This is funded primarily, what I showed here is funded primarily by two NIH R01s, one to me and one to Koff and myself. There's a lot of other people. I'm listing just the people that have worked on the computation. There's also a lot of people that have worked very hard on the biological assays as well. And with that, I just want to thank everyone for their attention.